Perfect. So good morning, everyone, and thanks for coming. This is the one before the end of this year's SEM seminar. We have one more last Friday, so don't think we're still on for holidays. I wanted to make a small announcement, like you can find the agenda of the seminars at the bottom of the email that you usually receive. There you can check who has spoken, who is going to speak, and you can have links to the YouTube videos of the seminars that have, of course, have happened. And one short announcement that we are now, probably have also received, we are open for you to invite new speakers. So we are expecting from you until December 19th, if I'm not wrong, right, to send your propositions for new speakers for the coming period, meaning from March to October 2026. Today we have Pleuni Pennings, and I'll give the floor to Emmanuel from Hopper to introduce here.
Thank you very much, good morning, everyone. I'm very excited to introduce Pleuni Pennings to you. She has joined ESEM and our team this year. She did her PhD in Germany roughly 20 years ago, right, with Joachim Hermisson, and then did a couple of postdocs, among others, at Harvard and at Stanford before being a professor at San Francisco State University. That happened in 2014, and you were a full professor before moving here, so that's a new exciting episode of your life. Some of you might know her from her work on soft sweets, but today she's going to talk about HIV and drug resistance, topics that she's covering today.
Thank you. And thank you, everyone, for being here. I know the end of the year is busy, and so I'm happy that you found the time to join us here. As Emmanuel was saying, I moved here recently. Is this working? No? It was working before? Oh, I don't know what this is.
I think you just have to click on the presentation.
Is it? Oh yeah. Perfect. That's okay. So I was in the United States for a long time, but originally I'm from the Netherlands. In San Francisco, I worked at San Francisco State University, which is a university that is mostly focused on teaching. And so I was always teaching a lot and trying to do my research alongside, just like many people here do that. This year, for many different reasons, I decided to leave the United States and come here. And one of the exciting things for me here is that even though I do not have a permanent job yet, I've been hired by the university for the next 3 years, but I don't have to teach, and so it feels like I'm a postdoc again, and I can focus on my research, which I'm very excited about. So today I want to tell you about.
You just need to click again.
I need to click here again.
Each time you select the other stuff, you have to click back on the presentation.
So now we're okay. I see it. Okay, so I want to tell you about the reversal of drug resistance. So when drug resistance, either antibiotic resistance in bacteria or anti-malarial resistance in malaria or antiviral resistance in viruses, when that has reached a high level, either within a host or in a population at large, is there a possibility for us to make the frequency of the resistant type go down over time? And one of the reasons I got interested in that is because I was doing a project that was actually focused on the stability of drug resistance. So this, let's see. So this is ciprofloxacin or fluoroquinolone resistance in E. coli in different European countries. It's data from the European Center for Disease Control. They have a lot of surveillance data, which is really cool. Each line is a country. And so you can see that here in the 2000s, resistance slowly went up. But then here, most of these lines are fairly flat, and resistance stayed at a stable level. And so I was doing this project on why there is this stable level. But then one of the things that I realized when I dove into the data more is that when you focus on this last part, and it's not very clear here, but these lines are actually going down. So if you blow up that last part of the plot, you see these lines like this orange one is Spain, sorry. And then this red one is Germany. This light green one is France. And all of them are going down slightly. And so I got interested in understanding what made resistance go down. And it turned out that if I look at the 20 largest European countries, in 16 of them, over those last 10 years, from 2011 to 2021, that's where I have the data on the usage of the drugs. In 16 out of these 20 countries, the usage of fluoroquinolones, per person in the country, went down. And in 15 out of these 16 countries where use of the drug went down, resistance against the drug also went down. On the other hand, there were 3 countries where over those 10 years, the country as a whole per person started using more of the drug, and in those countries, resistance went up. So here I plot quinolone use on the x-axis and percentage quinolone-resistant isolates in E. coli on the y-axis. And these are the countries where usage of the drug went down and resistance also went down. Again, this green one is France. And then there's these countries where resistance went up. The usage of the drug also went up. And then there's one country, Sweden, where sort of the unexpected app happened in the sense that the country used less drugs, but resistance actually went up. And then this little dot is the UK, where nothing changed. Okay? So this, to me, this made me happy, because it showed that if we use less of a drug, which a lot of countries and hospitals and doctors are working hard to try to reduce how much we use of certain drugs, right? How much we use of antibiotics, especially. If we do that, it looks like it's actually working, at least in this case. You use less of a drug at the country level over a couple of years, resistance goes down. So that's cool. So it made me interested in looking for other cases where resistance levels went down and trying to understand, what the, when that happens and when it doesn't happen and what we can learn from it. So why, apart from just my personal interest, why would we be interested in the reversal of resistance, or the lowering frequency of resistant types? Well, first of all, it improves patient outcomes, right? If, instead of 30%, 20% of E. coli is resistant to ciprofloxacin, then it's more likely that when I get a treatment for urinary tract infection or for something else, it's more likely that it will work. Also, in some cases, drug resistance levels are so high that countries decide to no longer use a certain drug. And so then, in that case, if resistance levels go down, maybe we can reuse these drugs. And then finally, I think this is just like an intriguing evolutionary system, and we can learn about things like compensatory mutations and linkage and selection and time scales and those kinds of things that sort of population geneticists are interested in. Okay, so I'm first going to tell you about this story about HIV and what happens within a person. I've spent many years studying HIV within hosts, and then I'll talk more about what happens at the population level. So one interesting case of reversal of resistance is this case in HIV where someone who is pregnant and HIV infected back maybe 20 years ago or 25 years ago, in many poor countries, there were no good treatments available for everyone. There were good treatments available, but there was not enough money to pay treatments for everyone. And so many countries had like these rules that you had to be like sick enough to become eligible for the combination therapy that we know works well for HIV. And so when someone was pregnant, oftentimes during pregnancy, the baby is actually not HIV infected, even though the mom is. But during labor is like a risky time. It's a time where the baby can get infected. And so what they would do is when the pregnant person would come in because labor had started, maybe the waters had broken, they would give her a single dose of nevirapine at that moment. A single dose of nevirapine, this is a very powerful HIV drug, and it has a long half-life, and it has very good penetration in the sense that it goes through the entire body. It goes into the baby as well, and it works as a prophylaxis for the baby. And it reduces the risk that the baby gets infected by about 50%. So it's not foolproof, but it's still very useful, and it saved a lot of babies. But what happened inside the body of the mother is that resistance mutations in the virus quickly increased in frequency. So this drug, even though they maybe only took one dose, sometimes actually they took 2 doses, but oftentimes it was just 1 dose, it stays in the body for about 2 weeks. So for about 2 weeks there's selection for resistance. And these resistance mutations are often already present at a very low frequency, and so they increase in frequency rapidly. Okay, so now, baby is born. At some point, this person, the parent, will become eligible for triple drug therapy. And it turned out that if you start triple drug therapy, which is 3 drugs, but it includes that same drug that they got 1 dose of. And so now, if the virus already has the resistance mutation against one of those 3 drugs, the probability that the whole treatment fails is much higher than if they had never had this single dose of nevirapine. So here's some data from sequencing data from the virus inside a person, within a host, inside a person who had taken a single dose of nevirapine. So after about 6 weeks, the median frequency of the resistance mutation is about 8% in this data set. I think that like if you would test like a little bit before 6 weeks, it may be a little bit higher, but I'm not sure. But if you wait until 3 months, it goes down. The median is only 1%. If you wait for 7 months, the median becomes undetectable, basically, and then at 12 months, it's undetectable in even more people. Okay, so the single dose of the drug makes the resistance mutation increase, and then it slowly decreases again. And then if you look at treatment outcomes, there are other people who did this kind of study. When women who had taken a single dose of nevirapine, they're starting ART, this is antiretroviral therapy, in this case it's combination therapy with 3 drugs. If they start less than 6 months after giving birth, so less than 6 months after taking that single pill, those who had taken the single dose had almost 50% chance of the therapy failing, meaning that the virus was replicating even though they were taking drugs. On the other hand, in the placebo, you did not see that effect. But if they waited more than 6 months, the placebo and the women who had taken the single dose of nevirapine, they did equally well. Or equally badly, actually. They do worse than these placebos. I'm not sure why that is the case. Okay, so it turns out that in this case, if you are able to wait for that resistance mutation, that frequency to go down again, if you're able to wait at least 6 months, that mutation sort of disappears and becomes unimportant again. And the treatment is much more likely to work well. And obviously that's really good for the person who's taking the treatment, right? Because we want the viral load to go down so that your immune system stays healthy. So this is a case where the reversal of mutation happens inside the patient and it directly influences the health of the person. Nowadays, this is no longer done because nowadays, well, actually, I'm not sure in 2025, but in 2024, before Trump started to change everything, treatments were available for most people, and especially pregnant people. Like the triple drug treatments were available for everyone, and so they didn't have to take the single dose anymore. Okay. So I'm going to switch to population level. So what happens in a country or in a city or in a hospital, so not inside a single host, but when we include like the between host situation. So I tried to find the simplest cases, at least as a start to see if I can understand what is happening. In this case, I tried to find cases where a drug was no longer used. So a country, or I guess in this case it's always countries, a country decides to no longer use a drug, and then I want to see what happens to the level of resistance in that country, the frequency of the resistance type in that country. Okay, so this is a very well-known example. It's chloroquine resistance in Malawi, in Africa. So chloroquine for many decades, the 60s, 70s, 80s, chloroquine was the most common treatment used against malaria. And then resistance spread first in Asia, and then it came to Africa, and it spread through the continent, and chloroquine was no longer useful. And in Africa, Malawi was the first country to change the treatments that they used as a first line, as the first choice of treatment against malaria. So in 1993, someone did a small clinical trial that showed that chloroquine did not work anymore in the majority of the cases, and then the country decided to no longer use this drug. And then 10 years later, this paper came out that showed that the resistance, the mutation that causes resistance, went down, very predictably over those 10 years. And when they did a clinical trial at the end of those 10 years, and they treated people with chloroquine, it worked in all of them. Okay? So in this case, it's very clear, you stop using the drug, there's no longer selection for this resistance mutation, and the resistance mutation goes away. And now in Malawi, malaria parasites are no longer resistant to chloroquine. Okay. A much newer example, also in malaria, came out earlier this year, a drug called piperacin. This is in Vietnam, where they decided to no longer use piperacin because resistance levels were so high. Resistance was at about 90%. They stopped using it, and resistance levels dropped very quickly. And in about 2 years, resistance was almost gone. Okay, so similar situation, but it was much faster. In bacteria, there are some of the same examples, but in bacteria, as far as I'm aware, there are no examples where countries decided to stop drugs for humans, but there are good examples where countries decided to stop certain drugs for animals. I guess it's easier to do that because the drugs for the animals are often used as growth hormones. They're not used to actually treat sick animals. And I guess even if it was used for sick animals, maybe it'd be easier to decide that the animals could be sick rather than deciding that the humans could be sick. Okay, so this is in China. Colistin resistance was a big problem in China, and colistin was used for pigs. And then when they stopped using it, resistance levels went down quickly. These are colistin levels in E. coli in pigs before the ban. It's about 45% on average across different parts of China. Then, when colistin is banned, a few months later, resistance levels are already lower, and then the year after it's even lower. I've not found any data for later than that. And some older data, much older data from chicken farms in Denmark, where there was a glycopeptide drug. They stopped using it, and there's this beautiful curve, I love it, where resistance levels, go down quickly and reach, not quite, but nearly 0% after a couple of years. So it looks like in these cases, there must be a cost to the resistance, a fitness cost for the pathogens, for the bacteria or the malaria, because when we take away the drug, resistance levels go down. It doesn't always work though, because there's cases where where drug levels, the use of drugs was much reduced, even though it wasn't entirely at zero, and it looks like there's no real effect. For example, this example, I think had, I've known about this example for a long time, and it always made me think that reversing resistance was not possible. This is trimethoprim, the use of trimethoprim in a county in Sweden. So here they did like this big study where they took 2 counties in Sweden, and in one county they encouraged doctors to not use the drug anymore, and in the other county they didn't change anything. And they did that for 2 years. And so reduction of the drug use was about 85%. So that's what you see here. That's a reduction in drug use. And then this is the result in terms of resistance. And there's like no effect. Now when they compare to the other county where they did not stop using the drugs, it looks like there's a little bit of an effect, but it's very subtle, and it doesn't look anything like the ones like the other examples where resistance levels go down drastically. So it's not clear why it didn't work here. I could imagine that the county is just not big enough in the sense that maybe there's a lot of migration of resistant strains into the county, and just reducing the drug level in a small area may not have a big effect if that area is very much connected to other areas. Another option could be that this particular resistance does not come with a cost, but that's really hard to test. Lots of researchers have tried to test for costs in the lab, but I must say that I'm a little skeptical of that, because I think if you do not find a cost in the lab, that does not mean that there's not a cost in real life. Okay, lots of hospitals have done intervention studies where the entire hospital worked together to try to reduce the usage of certain drugs and then see if it had an effect on resistance. And the results are sort of all over the place. Sometimes it looked like it worked, sometimes it doesn't look like it worked. Here, this is from a fairly recent review paper, and the conclusions are we could not confirm that restricting carbapenems or third-generation cephalosporins leads to decrease in prevalence of antibiotic resistance. But then they think that fluoroquinolone and piptas reductions may decrease resistance. So there's different of these sort of review papers, and all of them sort of say like, yeah, it could work, it may not work, we're not entirely sure. Okay, and then finally, this is actually at a country level. In this same study, this study was the study in Vietnam, where piperacin resistance went down so quickly and so beautifully. Chloroquine has not been used for malaria in Vietnam for a long time, as far as I understand, but resistance is still really high. This is from the same paper. And so in this case, you know, we cannot argue, I think, that there's no cost, because we see that in those African countries, the resistance did go down, the same, and it's the same mutation. I don't think we can argue that the area is too small, because it's a country, and this also happened in the same area. So I'm not sure what's happening here. And but one thing that I think may be happening is that actually chloroquine may still be used, even though it's not officially used for malaria. Chloroquine is also used for treating other diseases. And it also may be that there is like a black market kind of availability of chloroquine. This is really hard to figure out. There's one really nice paper by people who tried to find out in all these countries in Africa how much chloroquine were people actually using. So they did like these, they analyzed data from huge surveys where they asked people like, has your kid had a fever recently? If your kid had a fever, did you give it medication? If it was medication, did you give it chloroquine? And then they tried to compare like how much chloroquine was actually used in a country versus how much chloroquine resistance there was. And their conclusion was that in countries reporting sustained chloroquine use, chloroquine-resistant malaria persists. So they argue that even in countries where maybe officially chloroquine is no longer used for malaria, in reality it is still used, and that is what makes that chloroquine resistance is still at a high level. But this kind of data is really rare, and so I'm not sure how much we can learn about those like cases where it looks like resistance should have gone down and it didn't, because there could be so many reasons why that happens, and you really have to be lucky to have this kind of data to try to understand it. I'm going to check real quick what the time is.
Sorry. You have time. You have 15 minutes.
Okay. Okay. But this chloroquine resistance case, actually, I do think it's really interesting. So again, this is chloroquine resistance in Malawi after the drug was banned. You see this reduction in resistance. Later data show that resistance became undetectable. Many studies do not see any resistance. Later, the same happened in other countries that also banned chloroquine as a malaria drug. And it's not just that this is data for like a specific mutation, but we also see that chloroquine efficacy as a drug rapidly increased. So here in 1993, chloroquine efficacy was 17%, meaning that if you're infected with malaria and you take chloroquine and then a couple of days later you do another blood test, you see that the malaria is gone. Well, that only happened in 17% of the people in the early 90s, but then these newer studies, after the ban, show 100% or 99% efficacy of the drug. Okay, so it's not just that the mutation disappeared, it's also that we actually see that the drug works again. So I'm thinking, if the efficacies of these, if the efficacy of this drug is so high, and chloroquine is a cheap drug and has few side effects, you'd think that they want to use it again. But it's not been reintroduced. And I think And the worry is that if it was reintroduced as a malaria drug, that resistance would come back real quick. So for example, in this Laufer paper, she writes, it may become possible to reintroduce the drug in combination with other drugs to prevent the reemergence of resistance. So the worry is that if you reintroduce it by itself, you'll get resistance again quickly. And I don't think it would come back that quickly. Or it may not come back at all. So this is what I'm thinking. So here on the, oh no, what did I do? Okay, so here on the x-axis is how much chloroquine is used in a country. Okay, so here on the right is like chloroquine is used as the main drug, and everybody who is treated for malaria will get chloroquine. And here on the left is like nobody's using chloroquine, other drugs are used to treat malaria cases. And on the y-axis is the fitness of the resistant type relative to the fitness of the susceptible type. Okay, and so this line is the fitness of the susceptible allele, and if the resistant type is more fit, its fitness will be above that line. If it's less fit, it will be below that line. Okay, so when chloroquine is not used, we know that the resistant allele is less fit, right? That explains why resistance goes down in those countries where they're no longer using chloroquine. We also know that if chloroquine is used, the resistant type is more fit than the susceptible allele. I didn't actually show you any data on that, but there's, first of all, we know that resistance spread through Africa quickly in the 70s and the 80s and the 90s. And then for a few cases, we actually have data on like how fast it spreads in a country when chloroquine is used. So I'm pretty sure that the fitness of the resistant type is higher than that of the susceptible type when chloroquine is used as the main drug. And so we don't know what is the situation here in between, but we can imagine that the more chloroquine is used, the higher the fitness of the resistant type, on average, in a country. And so as long as we stay below some level, and of course we don't know currently what level that is, but as long as we stay below some level of treatment, there will be selection against resistance. And how much resistance that will result in will depend on how often the resistant type enters the country, either through migration or mutation, right? So because even if resistance has a fitness cost and is less fit on average than a susceptible type, if the country is like flooded with migrants that are resistant, you can still get like a fairly high level of resistance. And so for this particular resistance, mutation is probably not very important. And the reason I think that is because all of Africa, chloroquine resistance in all of Africa is due to an allele that migrated probably only once or a handful of times from Asia. Chloroquine resistance never evolved through mutation independently in all of Africa. And so that probably means that the mutational route is unlikely. But migration, of course, is still likely if there's other countries where chloroquine is still being used or where chloroquine resistance is still common, then migrants can enter. And so we have to think about what happens with selection and what happens with migration and how, and what kind of balance do we get between migrants that increase resistance levels and selection that decreases resistance levels, as long as we stay to the left of this line. So I realized as I was preparing this talk that this is actually something that I should calculate what is the expected level of resistance. And so this is like the simplest toy model that I could come up with, but I actually think it is something that I'm going to try to model more precisely. So here the idea is that Here on the left, chloroquine is not used, and resistance is very costly on average in the country. On the right, chloroquine is used a lot. Resistance has higher fitness on average in the country. And then, in addition to selection, there is some level of migration that could bring in resistant or non-resistant types. In this case, I made it symmetrical. And what you would expect is that resistance levels increase the closer you get to that level where resistance and susceptible types are equally fit. And then when the susceptible type is when the resistant type is more fit, it will quickly, the equilibrium level will quickly be at 100%. Right? So if we use chloroquine so much that on average chloroquine resistant is more fit than the susceptible type, then it will quickly reach nearly 100%. But as long as we stay at a chloroquine usage level that makes that the resistant type is less fit than the susceptible type, I think we can, I hope it can stay close to 0, even though it will not be exactly 0% because the migrants will always come in. So I think that it's worth trying to reintroduce chloroquine. And I think there's not much to lose because countries like Malawi, currently they do not use chloroquine at all. So if you reuse it and you find out that next year resistance levels are too high, you just stop using it again. You're already not using it, so you're already not benefiting from the fact that there is no resistance. One main reason why chloroquine I think should be used is because it's very cheap. So it costs about 10 cents per course, whereas the other types of drugs that are currently being used cost about $1.40. And so even if chloroquine is used for, say, only 5 or 10% of the people with malaria, it would save the country money. Okay, so what have I learned? Resistance can be reversed. It doesn't always happen, but there's really good examples where when we remove a drug from a situation, resistance levels will go down. And so it's not impossible, even though it's not guaranteed. And I think that when we actually look at the data, I think it shows that things are not as bad as the media often tells us. I feel like, when I read about drug resistance in the newspapers, oftentimes these are like doom stories. But in reality, I think resistance, we have opportunities to manage resistance, and actually some of the things that we are doing are already helping. Resistance levels are lower now than they were 10 or 20 years ago. So, my future plans, my plan is to stay at ISEM for now. The university has hired me for the next 3 years, so I'll be here at least for 3 years, but I hope it will become permanent. I'm writing application for the CNRS this month. In the spring, I plan to work on an ERC proposal. If anyone else is doing that too, I would love to share experiences. I'm also working on a book about drug resistance. If anyone would like to form a book writing or ERC writing club in the spring, I'd be very interested in that because I think together it's always more fun than alone. And with that, thank you for your attention, and I hope there's time for questions.
Oh, yes. Thank you, Pleuni. There is time for questions, and also I invite the people online to put their questions on the chat. So just raise your hand and just speak a bit loudly because the microphone is in the front.
I just have a curiosity question. How Malawi was so successful to just eliminate all the chloroquine use, even though other African countries seems to be using?
Yeah. That's a good question. How could it be that, like, in some countries, when the government basically says we're no longer using chloroquine, it works, and in other countries it doesn't? I'm not entirely sure.
No migration to Malawi. It says on the same continent.
Sorry, the same?
It's on the same continent.
Yeah. Let me show you where it is. I think Malawi is. Oh, there we go. This is Malawi, the skinny one. I'm not sure. I'm absolutely not an expert on this, but I think possibly Malawi is a country where it's a poor country, but it's also a stable country with a stable government and lots of external help. And so I think maybe all the malaria drugs in Malawi are paid for by the government, which in turn is probably paid for by the WHO or the United Nations. And so if the government decides to change, I think they can maybe change more easily than when people are buying their own drugs. That's a guess. If someone actually knows, I'd love to hear.
Is there any data on where the costs of resistance are paid? Within host, between host, so forth? Competition between strains?
Yeah. So for some of these resistances or some of these drugs, yes. For some of them, no, I don't know. There is, for example, for ciprofloxacin resistance in E. coli, people have tried to find evidence for cost within hosts and not found it. But then if you look at the population level, we see that resistance goes down when less of the drugs is used, so that is very suggestive of there being a cost, but we don't see it within the host, so it looks like it must be between hosts. There's one very cool example of a malaria drug. I'm blanking on the something. I'm blanking on the name a little bit. But in that case, they have shown that the cost is entirely paid in the mosquito. The resistant type of the malaria cannot survive in a mosquito. And so you get resistance, if you're treated with that drug, the malaria can evolve to become resistant inside your body, but then the mosquito cannot travel by mosquito. For the chloroquine case, I don't know. That's an interesting question. I don't know.
We'll have a question online.
So, Serafin Gutierrez, do you hear us? And you can talk normally.
Hi, do you listen to me?
Yes.
Yes.
Okay, perfect. Thank you very much. Probably this is a very naive question, but I was wondering whether you can use the rate of loss of the resistance along time in a population as a way to measure, to estimate the fitness cost associated to the resistance.
Yes, I think so. The question is about the rate of loss. Can you use it to estimate the fitness cost? And I know that people have done that for the malaria cases. So I think the estimated costs from different studies are maybe 10% per generation, and you have to make some assumption about the generation time of the pathogen. For the other cases, like for the E. coli and the E. faecium cases, I'm not sure if people have done that, but I'm sure it's possible, at least on a per time level, because I don't think we know the generation times for these things.
Okay, thank you.
Yes.
Yeah, sorry, I maybe have a little bit provocative question. Let's say you're going to write a book. I thought maybe give that some thought as well. I don't know what audience the book will have, but it seems that if I see what you're saying, that the far right is right, that reducing migration will reduce the cost of the health system in the country. So, can you have any thoughts or comments or on that?
Yes, well, obviously I have to be in favor of migration because I've been a migrant for the last 30 years or so. Let me see, is it always the case that migration, I mean, if we're worried about the spread of pathogens in general, whether they're resistant or not, migration is bad, of course. Like if we would all just stay home forever, not commute to work, not go to a cafe, and definitely not go in an airplane, you know, you would not get pandemics. But it would also limit the spread of all good things, like ideas and help, and so limiting migration in general, I think, would be a bad thing. But in some cases, limiting migration for specific things, like I could imagine that you encourage people to not travel when they have a fever. That would be a good idea, I think. But yeah, obviously, I'm not against all migration. That would be counter to my life.
You have to wait for the book thing.
Yeah.
I think there's another online question, right?
Yes. There is a question from Anne Boissière, who wrote it, so I'm going to read it. It's, what about bacteria multi-drug resistance, BMR? Is there an example of reversion?
Multi-drug resistance. So yes, I know that there are examples of like reductions of multi-drug resistance. For example, multi-drug resistance of tuberculosis has been reduced in Europe over the last 10 years or so. And I think for some of for maybe E. coli or Staph aureus, I think there are some cases too. I haven't paid too much attention because it's a little too complicated for my brain. Like when there's like multiple drugs, and then that makes it more complicated. And I try to focus on the cases where I feel like we can understand it better. But I do know that there is data where we see that multi-drug resistance goes down too.
So you think that understanding the one resistance cases would then help to understand better the multi-resistance case, that the processes should be linked or something?
I would think so, for sure. But of course, like, it could also, like, well, there could be several things going on. One is, if the resistances are independent genetically, then if resistance to one goes down, then whatever you call as multi-drug resistance will also go down automatically just because of how you keep track of it. But if there's actually, like, for example, a plasmid that has multiple resistances on one plasmid, then actually that could be a reason why you remove one drug, but the plasmid is still selected for because of the other drugs. And so it just means that there are more options, so it's more complicated, but I do hope that, like, by studying the simple situations first, we can understand the more complicated situations later too.
Okay.
I saw that there was a question there.
Yeah, Milan? Yes.
Yeah, thank you very much. I wonder, when you talk about resistance, is there only one resistance allele? Because I'm surprised that there is not a lot of different resistance allele with different costs.
Yeah.
In the population, like we saw for mosquitoes, for example?
Yeah, so in the mosquitoes case, in chloroquine resistance, it's almost always due to this one mutation, K76T, in the CRT gene. It looks like it's always the same thing. But in other cases, there are, in the case of the bacteria, for example, there are more options, and in the case of in some of the other malaria drug resistances too, there's also multiple options. I have not seen, I guess the interesting example here would be like if say you take away the drug and there's maybe two types of two resistance alleles, one of them goes down and the other one doesn't, or one of them goes down faster than the other one. I'm not aware of anyone having seen that or shown that, but I'm sure it must be possible. Yeah.
And another question for the treatment therapy, when you show that if you treat with the first drug and quickly after the treat therapy doesn't work as well, the treatment doesn't influence the resistance in the whole population. I don't understand why it's a within effect and not the selection of the population.
Oh, for the HIV case?
Yeah.
It's because, well, some of these mutations, they can also be transmitted to other people. But most of the time, once someone is infected, they do not get reinfected with HIV. So most of the time, once you follow an individual patient, it will be like one virus with one person forever. Yeah.
So I have a follow-up on all these questions, actually. To what extent do you think that science really influences drug use policy? Because what you showed is that when resistance goes up, there's an alternative drug that's used. Scientists follow whether or not resistance goes down, but there's not necessarily a policy that reintroduces the drug. And there's so many intricate, interesting things about the transmission and the costs and so forth that seem to go unused. Are there cases where science really influences policy? Because you're proposing that, but
yeah. I mean, I guess it depends a little bit on like what you call science or not. Like a lot of these surveillance studies, I do think they influence policy. Lots of sort of public health systems have they may have like cutoffs decided beforehand. It's like, okay, if in this area like resistance levels go above this, then we'll switch to this other drug. And so then surveillance clearly does influence science.
Right, okay. That's like risk management.
Yeah. I mean, I think that I think in HIV, there's definitely cases like where like the scientists, including like the people who were doing the population genetics of the within host dynamics of the virus, influenced how people were treated. At the same time, I think that clinical trials have much more direct influence on what happens. And I think there's definitely, this is why I'm doing this, I think there's more room for population genetics in this world. I think, sort of the stochasticity of mutation and like the nonlinearity of like how populations respond to selection and those kinds of things that population geneticists are used to thinking about, I think that often doesn't reach policy level. This is one of the reasons why I'm motivated to do this. Yeah.
Any other question? Yes.
I was wondering if resistance goes down very quickly, you could imagine that, it seems perhaps naive, but you could imagine that alternating treatments, treatment types could be a solution. Has that been tried or is it working?
Yeah.
Resistance
yeah.
go down for one treatment, but then
Yes.
still treatment.
Yes. I think that has been tried, but I'm not sure. There's been very clear results of whether that works. I do think that it makes sense to have, some kind of. So obviously, like one thing that we know that works in some cases is like combining drugs inside a patient, right? That's what we do for HIV, and nowadays it's used for malaria in most cases. But then, like, sort of at a population level, where they're like alternating or or saying like, you know, this group of people gets this drug and this group of people get this drug. Whether that works and when that works, I don't think we know exactly. But yeah, I know that it has been tried, but I don't know that there's been very clear results. And I think that's in part because we don't know necessarily like what scale we need to do this, or like how fast, or how big the block should be, or there's much to learn there, I think, still.
And I think there's one last question we take online.
So, Rémi, can you hear us?
Yes, do you hear me?
Yes.
Thank you very much for your presentation. Very interesting. Sorry to not be present in physical manner. So in bacteria, carbapenem resistance is usually possible thanks to plasmid horizontal transfers. So I presume we are expecting that it induces selective cost when the carbapenem are not used. Which is completely different from the quinolones that are point mutations. So bacteria becomes resistant to quinolones thanks to point mutations. So it looks to me that this is exactly the reverse of what you showed about the quinolone that you said, if I well understood, that it was coming from horizontal transfer from Asia. So on the bacteria side, we see that the carbapenem are remaining in the population, even though it's based on plasmids transfer. And on the other hand, you have the chloroquine that is disappearing, even though it looks
Even though it's a mutation?
yeah, and even though the chloroquine resistance is made by horizontal transfer, so that means it's clear a new gene that is introduced in the population, or how do you explain these two different outputs?
Yes. So I do not have an explanation. I agree with you. I think that we would expect that plasmids would be more costly than individual mutations. And so some of these results are maybe surprising. Chloroquine resistance is in malaria, which is a recombining eukaryote. So that's like maybe different. But yeah, I don't think we have enough examples to be able to say like, oh, mutations are more likely to be revert than plasmids. And of course with plasmids you often have that there's multiple resistances on the plasmid or other things also on the plasmid. And so it's harder. The plasmid may stay there because of other things than the resistance that we're interested in. So I don't think we currently know the answer.
Okay, thank you very much, Pleuni, and welcome. Thank you all, and thanks also for the people online. Perfect. Very clear. Thank you very much.
Thank you.
Yes.